Hey, welcome back to the Urban Monk Podcast. Dr. Pedram Shojai here with Martha Carlin, who is a microbiome researcher. She has spent a lot of time trying to figure out the connection between Parkinson's disease and the microbiome, and they've come up with some fascinating stuff. So, if you or anyone you know has Parkinson's in their life, this is a must listen. Tune in now. Well, it is certainly nice to see you. Welcome to the show, Martha.
Thanks so much for having me.
Thanks for doing what you've done. A lot of these healing journeys come through the work that's thrust upon us, right? And so you had a work to do on the home front to get here. So I'd love for you to share some of that story and contextualize this before we start geeking out on the microbiome.
Sure. So, actually, my education and background has nothing to do with the microbiome. I was trained a degree in accounting, came out and went to work for one of the big public accounting firms and trained as an auditor. And one of the main things they teach you is never take anything at face value, examine the evidence for yourself. And that really came in handy, has come in handy throughout my life. But in 2002, my 44-year-old husband, who was marathon running, healthy athlete, never done anything noticeably wrong that would have contributed to that. And I was like, how does a 44-year-old person get an old person's disease? I started to examine the evidence for myself and really look at what's flowing through the body. He was diagnosed with Parkinson's, and I really started to say, okay, well, what's flowing through the body? What are the main things that could be causal factors? But for about 10 years, I studied food and the food supply and what we are being exposed to through the foods and the chemicals that are used on the food, many of which are neurotoxic. And then in 2014, I read Dr. Martin Blazer's book, Missing Microbes, and learned of the term the microbiome. And it was just a huge epiphany for me because I was like, for a person who's an accountant, the microbiome to me seemed like the general ledger of the body, where it's keeping track, showing whether things are in balance. And later that year, the first paper was published that showed you could actually divide the two phenotypes of Parkinson's. One is tremor dominant and the other is posture and gait, where they're bent over like a penguin. And you could actually distinguish the two groups by the gut bacteria. And I was like, Eureka, that is it. And I quit my job and started funding some research at the University of Chicago with Dr. Jack Gilbert, and met my Suzanne Vernon, who had been studying chronic fatigue at the CDC for about 17 years. And the three of us came together and said, okay, what's holding back microbiome research? And it was good quality samples. And so I initially started out basically designing a kit to make it ick-free, to send the whole stool into our lab, and then we would process it into little aliquots and make it available to researchers all around the globe. And of course, we had a Parkinson's focus, but we had a broader focus because we could see at a more functional level that people were losing function, and it really presented as different diseases depending on where your weakness was. So we wanted to see, what are those common factors across people, and could we start to see differences in healthy aging populations, or, a baby microbiome, or all of that. And so for 7 years, studied the microbiome and worked with scientists around the world on some of their early research, and then went on to develop computational models of how microbes work together as teams and develop a bank of probiotic organisms, and then went on to develop a line of probiotics that specifically target lost functions in the gut.
So this is kind of where everyone's been hoping we'd get eventually, right? So Marty, when he first did his work, it was Papua New Guinea. They're looking at populations that hadn't been, they've been isolated and hadn't been exposed to our world. One of the original aha moments was, holy moly, look at the diversity of the species versus ours. And so that became kind of an operant play, right? For a long time, it's just like we need more diversity. We need more diversity means eat the fruits, the vegetables, the roots, the shoots, and the tubers to allow for those strains to be able to thrive, right? You can't have Honey Nut Cheerios and take probiotics and expect anything change. Now we're talking about something different. We're talking about this is boutique stuff now. We're talking about isolating strains that have specific functions. So tell me about that gap and what's happened in this last decade or so that is getting us to that specificity.
Well, there's now literally thousands and thousands of papers connecting all these different diseases to what's going on in the microbiome. But a lot of the early research was using something called 16S sequencing that is not really functional based. It tells you who's there, but not what they're doing. And what we did initially was we did whole genome sequencing. So we had the functional data from the beginning, but you've started to see a lot more metabolomics data. So looking at what the functions of the gut are, what they are producing. You've started to see more work around toxins and toxic burden. One of the areas that we also looked at was the toxin producing genes and toxin and antitoxin genes in the gut, because many of the pathogenic bacteria produce what are called pore forming toxins. And so those can form pores in our cells and spill out potassium and calcium, causing dysregulation. And so just from the body of research as it's expanded to really look at function as more of a critical factor, that's what we were looking at. Can you put together a team of microbes? Because in our body, it is a team of microbes that can shift the way you metabolize sugars. That's one of our main ones. Or can you put together a team that will help support the production of GABA, can help you with bacterial melatonin and tryptophan for sleep? Whereas in the earliest research, they were looking for single strains. What's this single strain do? How is that any different from a single drug? We're a complex system. You've got to start to look at the system and how you can bring in a SWAT team, if you will, guys that can work together to replace what you've lost.
So this whole terrain-based conversation has been going back and forth for a while now. Do you go in there and do you clean up the bad players, the LPS producing gram-negative guys? Do you have to do some work on the front end before you introduce your SWAT team? How are you finding the sequencing to work?
I'm not a practitioner, but what I will say is our base formula that we made, I actually made for my husband. It came from going to a meeting, the World Parkinson's Congress, and there were some researchers there showing that the sugar alcohol mannitol could stop the aggregation of the proteins and pull the aggregates out of the brain of this mouse. And I came back and I got a little mannitol chemistry book and started studying mannitol. And in that book, there were a handful of microbes that actually convert glucose and fructose into mannitol, which we don't use, but the gut microbiome uses. So beneficial bacteria can use mannitol as a carbon substrate, but many of the pathogens can't. And so I gave it to my husband. He had some pretty remarkable results. The UPDR score, which is the Uniform Parkinson's Rating Score, went from a 35 down to a 20, and the higher the number, the more advanced you are. And it stayed there for about 4 years until he had COVID. So he was very stable. And so we started to look at that. We wanted to do a clinical trial in Parkinson's, but there's really no agreed upon biomarkers. And we knew that it shifted metabolism. Well, in Parkinson's, about 40% of people with Parkinson's have insulin resistance. And so we did a clinical trial in people with diabetes, and both diabetes and Parkinson's have an LPS endotoxin model for inducing that in an animal. And so I said, well, let's look at serum endotoxin in the blood and endotoxin in the microbiome and see if that moves as a part of this. And it actually was one of the most significant shifts was the reduction of this endotoxin. So you asked, do you start by getting rid of those guys first? Well, by changing the ecosystem, we got rid of those guys instead of the killing approach that a lot of people attempt to do with microbiome modulation, whether that is herbally or using antibiotics. Those have downstream effects too. So we shifted the terrain by bringing in the police, if you will.
Yeah. Yeah. The biofilm starts to push things out. Have you looked at gut permeability in all these cases to see if the gut permeability has not been resolved, if there's zonulin, occludin, those numbers are skewed? Does that influence how this works?
We haven't looked at those specific markers, zonulin, but the endotoxin is crossing over that gut barrier. So just by virtue of
You know what's
having
happening?
significantly lowered endotoxin in the blood, you know it's happening.
Yeah. Yeah. So by theory, then what's happening is the good players that you're introducing are effectively shoring up the colonies and helping patch up a leaky gut because the body gets a fighting chance to heal itself, right?
Yes, and I think there's a couple of things going on. So one of the things we could see is that the gut was returned to an anaerobic state. So a lot of the pathogens thrive in an aerobic environment where there's oxygen, but some of those really beneficial bacteria like bifidobacteria, they cannot tolerate any oxygen. So we returned the gut to an anaerobic state so that bifidobacteria could flourish again. And the other thing is one of the strains that's in the formula is actually a strain called Leuconostoc mesenteroides, and it's found, I won't say universally, but almost universally in fermented foods around the globe. And it's a first colonizer in a fermented food where it removes the oxygen and acidifies the environment for the lactic acid bacteria to then come along. And it's also what's called an exopolysaccharide producer. So it produces what I would say is kind of like an artificial mucin, if you will. And this is just my theory, but is that perhaps those exopolysaccharides, one, they're feeding some of the other good bacteria, but perhaps it's also soothing the gut lining.
Mm. And sucking the oxygen out of the environment, helping bring up the acidity and letting the good guys thrive again.
Yeah.
That's interesting. So what your husband, we kind of stopped the story at him getting COVID. What did COVID do?
He had a very, very severe case of COVID. We had about 2.5 years of really working to try to get back from the damage that COVID had done. But a year ago, September, he actually had a pulmonary embolism and passed away. As a part of the work that we've done, we've come to really start to look at what's called the glycocalyx, the fuzzy layer that surrounds our cardiovascular system, all of our cells, our blood-brain barrier, our gut lining, and that damage to the glycocalyx is perhaps part of the Parkinson's pathology, but COVID itself does a lot of damage to the glycocalyx. That has some pretty severe long-term implications if you can't get that back in order.
Yeah. Well, I'm sorry to hear your husband passed over this. I'm hoping his legacy is going to help a lot of folks with the work that he drove you to do. So what are we now seeing? You said there's two subclasses there, the penguins and the fidgeters. Is there a different path on the probiotic journey for the different classifications here?
That's a really interesting question. There's a new book out called The Parkinson's Plan by Dr. Ray Dorsey, and a lot of his focus has been on environmental toxins. And it doesn't go into the real connections to the microbiome, but if you look at the toxic profiles and you look at the microbiome and you know what microbes can degrade what chemicals, you get an ecosystem that is paired with the environmental exposures of your lifetime. And that's one thing that I was looking at from the beginning also with my husband. He was raised in Cleveland, Ohio when the river caught on fire. So lots of air pollution, water pollution. He worked on a golf course throughout high school. So lots of chemical exposure there. He was a caddy. So out on the golf course every day. He worked in the oil industry out in oil field services. So lots of exposure there. And then he ran a men's clothing store, a high end, wearing a suit that was dry cleaned every day, and that's actually one of the key focuses of Dr. Dorsey's book is looking at dry cleaning fluid. So each one of those things is assault on our membranes, a shift in our microbiome, and there's now two camps, body first or brain first. And that may be related to whether it's the nasal microbiome and something that comes in through the nose, or the oral microbiome or something that comes in through food and colonizes the gut. But I think we're going to show, we have a paper hopefully coming out before the end of the year that's going to show how, regardless of whether it's colonization through the nose or the mouth, that the mechanisms are very similar. And part of it is driven by that battle in the gut and what it's doing to our cells and to the good bacteria that's spilling out that potassium and calcium and short circuiting the signal to the brain.
I mean, there's so much conjecture. Everyone wants their model. It doesn't matter who's right. It matters who we're helping, right? And this idea that, okay, nasal versus mouth versus the pathways, is it the toxins? Is it the microbiome? When you look at a client and you do an assessment, you're looking at maybe organic acids, you're looking at common toxins, glyphosate, I want to get into in a minute. But does it make sense to radically detox first because some of these people are so compromised, or does it make sense to build the terrain and detox simultaneously? I'm just curious how you phase this.
Well, I would say for a practitioner, detoxification is important, but also people with Parkinson's are very sensitive. Like, you know how most of us can wobble to one side or the other and be okay. I think as the Parkinson's advances, the sensitivity or the ability to wobble at the center becomes much tighter. So the process of detoxification, I do think it is important because if you don't remove the toxic burden, it's very hard to restore the terrain. You've got to go about that very, very gently. Um,
That's always been the challenge is you
yeah,
detox too fast, you make people sick, the art and the science of it all. We bought this beautiful house on a golf course. Got a great deal. And it was this beautiful house. My wife loved it. Everyone loved it. And I didn't know enough at the time. And all of a sudden, I'm like, what are they spraying over there? My white dogs had blue bellies. And I started looking into the use of this neurotoxic, cancerous, nasty sludge that they use on golf courses. And so your poor husband was a caddy. We actually signed a film a while back following this story and how so many people get these weird esoteric cancers attached to golf courses. So what do we know about glyphosate? What do we postulate about glyphosate? It's really hard because there's a lot of misinformation being chucked out there too to protect the industry. But glyphosate, there's a lot of smoking gun around this.
that's been a particular passion project of mine for a very long time is trying to understand all the different ways that it is causing problems. One of them that I'm actually surprised I don't hear any people really talking about. You do occasionally hear people talk about something called the shikimate pathway because that is the target of glyphosate, but they don't talk about what the shikimate pathway is. Something humans don't have, but plants have and microbes have. There are three amino acids that we can't make that we have to get from a plant or an animal who has eaten a plant, or we have to have microbes in our gut make it. And that tyrosine, tryptophan, and phenylalanine. And those are really essential to a number of our key neurotransmitters. And I have this, I'll say a little book, it's probably 200 pages, but there's 3,900 secondary metabolites that are downstream in that shikimate pathway. And dopamine is one, melanin is one. All of the steroid hormones are in that pathway. So when people start to have issues with these secondary metabolites, I don't think anybody's really looking at the connection to glyphosate, but glyphosate is a minerals chelator. And the trace minerals are really essential to enzyme function in the body. And as you bind up those minerals in the soil, the plants are not as nutritious. Then we're not getting the micronutrients we need. So then we have problems with enzyme function. And you have all these people now who have to take enzymes because their enzymes aren't working. or people who are mineral deficient, because our food supply is mineral deficient. And that's because we're spraying 400 million acres across the US multiple times a year with something that's binding up these minerals. And some of it does degrade to some extent, but not when you're consistently spraying it over and over again. And I've actually done quite a bit of work. My chief scientific officer, Dr. Raul Cano, founded another company called Ancient Organics Bioscience. And for the last 6 years, we've been working on soil probiotic formulas that can actually break down glyphosate, increase the nutrient content, and increase the yield by restoring and unlocking those bound minerals out of the soil.
Now, is this a product that goes to farms that are converting back from conventional to organic? Is it for cross spray? Well, who's using this? Who's buying it?
It's an uphill battle breaking into the ag market, I will say that.
Mm.
We do have people who are converting or attempting to convert. That's part of our audience. Some of it is people trying to test and see what it does and whether or not it might help them convert. And some of them are people who are, I've got to use this product in my farming, and I'd like to have a tool that helps me clean up after I use it. It's a little bit of everything.
Interesting. There's a lot out there. There's a lot of talk. The internet gets a little wild. People are like, well, you can't blame the gluten. It's the glyphosate. Well, Fasano proved that gluten also absolutely definitively tears the gut lining and zonulin levels are a thing. And there's a lot of either or 100%. But you look at systems biology, you look at the microbiome, it's an ecological model. What if it's gluten and glyphosate? What if it's gluten, glyphosate, and the heavy metals? People want to oversimplify and find a bad guy. Maybe it's all of it, right?
Yeah, and I say that a lot. And my husband used to say Parkinson's is a designer disease because people have one person has these 5 symptoms and one has these 3 and this one over here has these 2, and sometimes they overlap and sometimes they don't. And if you start to look at total toxic burden, all these different factors that fit in, it can also help you explain the variability in symptoms. And we've been in a model looking for a magic bullet for decades and decades. And you can't have a magic bullet for something that is complicated. You've got to understand that the body is an ecosystem, and we've been trashing the ecosystem pretty heavily with all the things that we're doing. And so, really starting to understand that and how to care for yourself like you are an ecosystem and think about all of these things and what a tiny little impact of each one of these things adds up to a pretty heavy total toxic burden.
Yeah, you get the world we live in. So someone's listening to this saying, okay, Parkinson's, that's really good. Thanks, Martha. But my mom's got Alzheimer's. My kid's got autism. How far has this spread into other diagnosable illness? Or are you just getting started on the Parkinson's side? Where's your research going?
So I do have a group of autism researchers who are actually looking at the Sugar Shift product, which is the one I developed for John, and they are looking at doing a study potentially in autism for that.
Mm.
I would say both Alzheimer's and Parkinson's have sometimes been called type 3 diabetes, so an insulin resistance of the brain. And so I do think there is potential for it there, but we haven't studied it there, and that's not on the slate right now. We do have a product slated for our next clinical trial, which is called the antibiotic antidote. And that one is really to try to restore the gut after if somebody has to take antibiotics. They're beneficial for acute problems. And so certainly we don't want to say never take an antibiotic, but if you do have to take one, there's often a lot of gut disruption afterwards. And so we're slating in a clinical trial to look at restoration of the gut with that product.
There's been a ton in the realm of FMT. Yeah, so fecal matter transplant. It's gross and people don't want to talk about it because it's poop. But there has been, I get a lot of studies that land on my desk showing just incredible results with people who's basically press and reset on their microbiome and taking healthy microbiome from someone else. What's your take on that? What's your take on global approach versus a more targeted strain approach? Although you're using multiple strains, you're still a family of strains.
Right. That research is really interesting to me. There, last year, I think, or it might have been the year before, there were two clinical trials in fecal transplant and Parkinson's, actually, one from the bottom up and one from the top down. And the one from the bottom up was not as successful as the one from the top down. And my personal thought is that a big part of the issue is in the small bowel in Parkinson's. And that is something you also miss in a lot of the microbiome testing. You're not going to see what's going on in the small intestine. But I do think we'll start to see more of that. And one of the big pioneers of that was Tom Borody in Australia. And he did, I want to say, probably 50 or 60 different people at different times with Parkinson's and had variable results. Method of delivery, how far advanced the internal ecosystem probably has something to do with that. And now we had approval, tentative approval for using FMT in C. diff, and they've backed off of that approval now, and that they have, I think, one or two standardized products. But I have talked to some GI docs who say those standardized products, they do not find that they are as successful at colonizing and basically sticking as they were having when they were maybe taking a household control, like somebody in the household who's been exposed to a lot of the same things, but they seem to be resistant to whatever the problem in the other person is.
Right.
Back in the early cowboy days of the FMT, when we first started collecting fecal samples, I had a young guy who had some pretty severe IBS, and he decided to do his own fecal transplant using his father's microbiome sample, his father's stool, and it actually made him much worse. I do always caution people if they think they're going to go do it themselves, not to do that without any kind of proper screening, because there's good and bad in there, and you've really got to be looking at that and knowing what you're putting into the system.
Yeah, I know they do a ton of front end screening now on the subjects, the donors, just to rule out some of that. But that's a novel treatment. It's very much in its infancy. This stuff is 30, 40 years ago, you just eat yogurt. And now look at what we're talking about. So there's a lot that has come. Where do you see it going? Where do you see in the next 5 years where some of these targeted therapies can be?
I would like to say that I would see it in the clinic, but over the last 10 years, there's been a lot of progress on the research, but not a lot of progress bringing it into the clinic. You do see it with functional medicine practitioners really starting to try to understand the importance of the microbiome and how to modulate that. But if you look at the vast majority of the population is still not going to a functional medicine practitioner, and the microbiome is nowhere near the doorway of most of the physicians that people are going to. They're not aware of it. They wouldn't know how to look at a report or how to even begin to approach a microbiome therapeutic. So I think functional medicine is going to stay at the forefront of that and start to show the evidence, but it's probably another decade before it makes it into clinical practice more widely because it's got to start being taught in medical school.
Yeah, I think you're optimistic in that actually. Yeah. Low turning wheel, and there's big business in between us and that reality, right? I'm a big fan of all of this. This is the future. The gut is absolutely the pivot for health. I've seen it in, tens of thousands of people at this point. And there are a lot of promises made. There's a lot of hyperbole. And there's a lot of yahoos that are just labeling something and saying this is the next greatest thing. So talk to me about the research you're doing on these strains. Talk to me about how this stuff's manufactured. Why is this different?
So while we have 4 strains of our own that we have manufactured, we have a whole bank that we'd like to someday do something with. And we're actually doing some work right now on a strain called Deetsia. So this strain that has been shown beneficial in cows in Johne's disease, which is basically the cow version of Crohn's. There's been a few small studies in humans where it has reversed Crohn's in about 3 weeks. We've done the safety studies on this microbe. We have 2 clinical trials set up to look at it in Crohn's, and we're in production right now small scale production. It's a difficult microbe to grow, but we have a company in Atlanta that is working on that with us. That's one of the ones I'm most excited about. But we have 4 strains that we initially started with, and that's the Leuconostoc mesenteroides that I mentioned. You don't find that in a lot of probiotics. You will find it in fermented foods. We have a Pediococcus acinolactici. That's often found in fermented meats. It's proteolytic, so it makes some enzymes that can break down proteins. We have a strain called Lactobacillus ruminus that actually came from a healthy human gut out of our sample bank. And that's a really interesting microbe. There's been some research, not on our strain, but another strain of ruminus, that it essentially prevents the attachment to the epithelial cells in the gut of pathogens. And what we found in our research was in a petri dish, basically, against Salmonella, E. coli, Listeria, knocks out all those pathogens. What you're looking for is beneficial microbes that can produce what are called bacteriocins, which is a very specific antibiotic that will knock out the bad guys, but not kill the whole environment. And so our Pediococcus has something called pediacin, and then we have a Lactobacillus plantarum that can break down glyphosate, but also has plantaricin bacteriocins that have a lot of targeted benefits against pathogens. And we have those made at a fermentation company in Wisconsin.
Amazing.
Specifically for us, yeah.
There is a lot of talk about the benefit of soil-based organisms. Go outside barefoot, go breathe in the forest, and there's a cumulative net positive effect of being on nature. Curious, topicals, transdermals, is anyone moving in the space of actually getting bacteria on the skin and not causing rashes?
So there are one of the earliest pioneers in the microbiome space was a company called Biome, and they had a product called Mother Dirt that you could spray on your face. And they ended up getting all these people saying, it's gotten rid of my migraines, my sinus problem, all these other things. It was a microbe called Nitrosomonas eutropha from the soil that they had identified from horses rolling around in the dirt. They went on to down a pharma path after people started telling them all of this, but they're actually back into the skin space. And then one of their early founders, a fellow by the name of Larry Weiss, founded another company called Symbiome, and they are working with the microbes from the Yanomami tribe and plant-based skin products. But there's not a lot of people in the skin microbiome space. There is an interesting microbe, Raul, who has the ag company. He has a microbe called Micrococcus luteus, and it's in our concoction for the soil, but it's found in the indigenous populations very much on the skin. Sometimes it's found on our skin, but it has UV radiation, lots of UV protection genes. It has what are called resuscitation genes. So if it dies, it can come back to life. DNA repair. There was a article in the Wall Street Journal maybe 2 or 3 years ago about some sunscreens that actually had Micrococcus luteus in it.
I just find it fascinating that the cosmetics industry has had its hooks in people for so long. We're talking about petrochemicals, volatile organic compounds, crap that makes you sick, neurotoxic stuff, but you put it on your skin because someone put an influencer on it. But some of this research, I find it fascinating that some of this research has been buried and not in the forefront. Is just get out in the dirt and things get better, right?
Yeah. I mean, I don't I personally, after learning so much about the microbiome, I don't use any products at all on I wash my face with water.
Mm-hmm. Mm-hmm.
And that's it.
Yeah, absolutely. I really appreciate the work that you're doing, Martha. I think that again, this is the forefront of where medicine is going in terrain-based medicine, functional medicine, and it takes pioneers like yourself. Unfortunately, you had a story that drove you into it, but you're helping a lot of people. Thanks to your ex-beloved who is, no longer with us, but his legacy lives on, right? And you're doing a lot of great work. So tell people where they could find you, about the company, the products.
Sure. Our probiotic brand is called BiotiQuest. That's BIOTIQUEST. urbanbook.com. I also write a Parkinson's blog. Some of it is about microbiome related, but a lot of alternative health, mindfulness, breathing, qigong, all of that. And that's called Martha's Quest. And then I have also a Martha's Quest Substack where I cover a lot of these environmental factors and some of the Parkinson's stuff.
Amazing. Well, thank you for your time, and I'll put links in the show notes for all your stuff so our people can get it.
Great. Thanks so much for having me today. I enjoyed our conversation.
Thank you. So did I. Well, I certainly hope you enjoyed that as much as I did. I'll put links for all of Martha's products in there in the show notes. If you're listening to this, just go to theurbanbook.com/podcast. Share this with somebody who might benefit from understanding that there's a lot more going on in the realm of functional medicine than what the conventional doctors are trying to throw at your loved one with Parkinson's. Share it broadly. Check out the products. I will put links to all of Martha's places you could find her in the show notes, and I'll see you in the next one. Thank you.